Photosynthesis in plants, one of the important chapter in plant physiology, 11th CBSE biology. This video related to the important points from this chapter which are going to come in the NEET exam. So students, this is the photosynthetic reaction where we can observe that 6 molecules of carbon dioxide, 12 water molecules in the presence of solar light and chlorophyll pigment involve in the formation of one glucose molecule, 6 water molecules and 6 oxygen molecules are going to release. Several scientists have worked on this photosynthesis process and they have given some important points related to this phenomena. Among that, Joseph Priestley, he conducted bell jar experiment and he proved that role of air in the growth of plant. Then John Ingenhousz, he discovered photosynthesis and he proved that sunlight is essential for photosynthesis and this process purifies the air. And Julius Vone, by photosynthesis pathway, he concluded that glucose is one of the major product of this process. Reuben, Camin and Hasid, they used O18 radioactive oxygen water and found that oxygen evolved in photosynthesis comes from water. The oxygen evolved in photosynthesis has come from water and they conducted this experiment on chlorella plant, chlorella algae. We know that chloroplast is a site of photosynthesis. In that thylakoid means grana involve in light dependent reaction which produce ATP and NADPH2. Whereas stroma meant for dark reaction or light independent reaction of photosynthesis where we can find the reduction of carbon dioxide in the stroma part of the chloroplast. And remember students, chlorophyll has a tadpole-like structure with a porphyrin head and a pylol tail. Remember, chlorophyll has porphyrin head and pylol tail, tadpole-like structure. And in the porphyrin head, you can find magnesium in the chlorophyll structure. And chlorophyll is one of the most abundant photosynthetic pigment found in all the plants, and it absorbs blue and red light and it transmits or reflects green light. Chlorophyll reflects green light and the accessory pigment is chlorophyll B. Accessory pigment is chlorophyll B. And another important pigment in plant is carotenoid, which is yellow to orange in color. And remember students, carotenoid is considered as a shield pigment. Carotenoid is a shield pigment. Why? Because carotenoid protects chlorophyll from photobleaching. Carotenoid protect chlorophyll from photooxidation. That's why carotenoid is considered as shield pigment as it is protecting chlorophyll from photobleaching. And remember, carotenoid absorbs violet and blue color of the visible spectrum. In chloroplast, highest number of protons, highest number of H plus ions are found in the lumen of the thylakoid. In the lumen of the thylakoid, we can find highest number of protons. Then during photolysis of water of photosynthesis, there is a release of oxygen in the presence of element manganese, calcium and chloride ion. Photolysis of water leads to the release of oxygen and that occur in the presence of elements manganese, calcium and chloride ion. And photorespiration, which is opposite to photosynthesis, requires 3 organelles in the plant cells. One is chloroplast, peroxisome and mitochondria. Remember this order: chloroplast, peroxisomes and mitochondria. Cyclic photophosphorylation, where photosystem 1 is going to involve cyclic photophosphorylation. In that, there is a formation of 2 ATPs in cyclic photophosphorylation. And remember, there is no reduction of NADP. In cyclic photophosphorylation, there is no reduction of ATP, and whatever the electrons ejecting from photosystem 1, the first electron carrier is ferredoxin. In photosystem 1, first electron acceptor is ferredoxin, followed by plastoquinone, cytochrome complex, then last is plastocyanin, and finally electrons come back to photosystem 1. Remember here, very important, electron acceptor is ferredoxin, first electron acceptor in cyclic or photosystem 1. PAR means photosynthetically active radiation. Remember, out of total incident solar radiation, the proportion of PAR is just less than 50%. Less than 50% of total incident solar radiation for PAR, photosynthetic active radiation. Remember these 2 points. And chlorophyll in thylakoid, chlorophyll in chloroplast located in the thylakoid. In the thylakoid, we can find this chlorophyll pigment. In the thylakoid of the chloroplast, chlorophyll pigment is there. In C3 plants, we can observe Kelvin cycle or C3 cycle, that is a part of dark reaction, light independent reaction. And in C3 plants, the first stable product is 3-carbon phosphoglyceric acid. 3-carbon phosphoglyceric acid. Whereas in C4 plants, in that C4 cycle we can observe the first stable product is oxaloacetic acid, 4-carbon oxaloacetic acid. That's why C4 name has been given. And this oxaloacetic acid has 2-carboxylic acid, 2-carboxylic group. That's why this C4 cycle is also called as dicarboxylic acid pathway. Dicarboxylic acid pathway. Okay. And remember children, C4 cycle, dicarboxylic acid pathway or Hatch-Slack cycle is the alternative pathway of carbon dioxide fixation. Usually in most of the plant, carbon dioxide fixation during dark reaction occurs by Kelvin cycle or C3 cycle. But C4 cycle is one of the alternative path for carbon dioxide fixation. Then maize is one of the best example for C4 plant. It shows Kranz anatomy in the leaf. Kranz anatomy. And remember C3 cycle, which is also called as Kelvin cycle, it has 3 important steps. First is carboxylation, reduction, and last is regeneration of ribulose bisphosphate. And in carboxylation, which is considered as the most crucial step, the carbon dioxide is accepted by ribulose bisphosphate. And in the last step, that ribulose bisphosphate regenerates to continue another Kelvin cycle. In Kelvin cycle, 6 carbon dioxides, 18 ATP and 12 NADPH are going to involve, and at the end of 6 Kelvin cycles, 1 glucose, 18 ADP and 12 NADP are going to produce. In C4 plants, phosphoenol pyruvate is the acceptor of carbon dioxide. In C3, ribulose bisphosphate. Whereas in C4, phosphoenol pyruvate accepts the carbon dioxide and produce 4-carbon oxaloacetic acid in C4 plants. And this is very, very important point. Remember, at higher temperature, photosynthesis decrease in C3 plant and increase in C4 plant. So, wherever high temperature is there related to photosynthesis, choose C4 plants. At higher temperature, photosynthesis is greater in C4 plants. And remember Emerson performed one experiment on chlorella. Emerson performed a photosynthetic experiment on chlorella and he discovered 2 photosystem, that is photosystem 1 and photosystem 1. Who discovered? Emerson, while working on chlorella. And children, rubisco is one of the most important protein on the earth, most abundant protein or most abundant enzyme on the earth, rubisco. Okay, and it's a bifunctional enzyme because in addition to accept carbon dioxide, it also accepts oxygen. That's why rubisco is bifunctional enzyme. Remember the important point. Usually photosynthesis are oxygenic, which photosynthesis means there is a release of oxygen. But there is a unoxygenic photosynthesis also we can observe in some bacteria. Anoxygenic, where oxygen is not released. That photosynthesis is called as anoxygenic photosynthesis. Example, rhodospirillum, which is a non-sulfur purple bacteria, chlorobium, chromatium. These are some of the bacteria where we can find anoxygenic photosynthesis. And remember, photorespiration is light dependent process. It occurs only in C3 plants. In C4 plant, there is no photorespiration. So we have to differentiate C3 plants and C4 plant based on the photorespiration. Okay, and photorespiration is opposite to photosynthesis because in photorespiration there is a uptake of oxygen and release of carbon dioxide. Okay, and C4 plant cannot perform photorespiration because of Kran's anatomy. And CAMP pathway in C4 plants we can observe crustacean acid metabolism that helps the plants to conserve water. CAMP, because of CAMP there is a conservation of plant, and also there is a closing of stomata during daytime. In this way CAMP pathway is going to protect the C4 plants. And Melvin Kelvin, he used radioactive carbon 14 in chlorella alga to study photosynthesis, and he discovered that first carbon dioxide fixation and the product is 3 carbon phosphoglyceric acid in C3 plants. Then remember, C4 plant has larger number of chloroplast. C4 plants has larger number of chloroplast compared to C3 plants. That's why C4 plants are more efficient in photosynthesis. Compared to C3 plants, C4 plants are more efficient in photosynthesis. In C4 plants, mesophyll cells carbon dioxide fix to form malic acid. And in photosystem 2, quinone is the first electron acceptor. In photosystem 2 or non-cyclic photophosphorylation, the first electron acceptor is quinone. Whereas in photosystem 1, ferredoxin. Remember, in C3 cycle, in C3 plants, to produce 1 glucose, to produce 1 hexose, 18 ATPs are required, 18 ATPs. Whereas in C4 plants, to produce 1 hexose, to produce 1 glucose, 30 ATPs are required. So compared to C3 and C4 plants needs 12 more ATPs to produce 1 glucose. So these are the important points from this chapter. Please revise. If you like the video, please subscribe to my channel. Thank you.